It's my pleasure to be talking about clinical trials, which is near and dear to me. And first, the question: which of the following are true about clinical trial participation? A, represents a generally more risky treatment option. B, is only recommended for patients that have no other treatment options. C, provides at least as good a treatment option as standard of care. D, opens up access to promising drug treatments that are otherwise not available. Or E, requires that once patients are enrolled in a study, they are obligated to continue the study even if they change their mind. So most people said 4, which is correct, opens access to promising drug treatments, and also 3, provides at least good a treatment option. But I think the other ones are important, that not everything is more risky. There are also studies for patients that have many treatment options. I always say that today's experimental treatment is tomorrow's standard of care. And lastly, there is not an obligation that patients must continue once they're enrolled. There's always the option to discuss with your healthcare team about either modifying the doses or coming off study. So what are the goals of clinical trials? We're trying to develop treatments and strategies to potentially lengthen lives, improve the way we use current drugs and regimens, develop new medications. Even though we've had a lot of progress and you've heard about those 10 drugs, the 3 groups that perhaps are still not yet medical are frail elderly patients, renal failure, and patients with high-risk myeloma. So we still, even though we've had all these drugs, we still need to do more work. And we also need better. As we were talking about earlier, the rational selection of drugs, when you have the alphabet soup that Dr. Martin went through, identify new targets and the next generation of compounds. And you've heard from Anne that the survival rates have nearly doubled, and we expect more improvements in the future. Shown here, when you look at the classes of drugs, are the circled in red are the 10 drugs approved since 2003, and each of those required a clinical trial. And many of the new drugs are being obviously currently studied in trials, and our hope is that we'll not be able to not only have more drugs added within each class and add new classes, but really be able to understand which classes, and equally important, how long. We still don't have answers to what myeloma. Should patients with smoldering myeloma be treated, and if so, when? What is the best treatment for newly diagnosed myeloma? Is it RVD, or can we do better? What are the best drugs and combinations for relapsed refractory? You've heard about a lot of triplets, but we keep getting unprecedented responses, as shown by the DARA-based first relapse studies. And then how can we match these patient subtypes in genomics or personalized medicine? So who should participate in studies? Obviously, the more patients that participate, the faster these questions can be answered. We need to accelerate drug development, help gain access to exciting new drugs. As I alluded to, the MMRC, MMRF have been instrumental in lobbying with the FDA to get these new drugs approved quickly, and the FDA is willing to hear new regulatory options, such as minimal residual disease, as a way of getting drugs approved potentially earlier. So a lot of work is being done on many fronts. So how do new drugs get developed? Most agents are tested in lab models. So first we look at cell lines. One of the limitations of this is that myeloma cells generally don't do very well outside the marrow. So the very fact that we have cell lines of myeloma cells growing and repeatedly growing and being what we say passaged, that's not a typical myeloma cell. So we're picking out myeloma cells that are really funky. The next step is in the animal model. Why do we do that? There was actually a question of why are mice used? And part of it is just historically, it's a relatively small animal, and we need to understand, even though the drug may kill the myeloma cells, what good is it if it has profound toxicity? And so you really need to understand not only the benefit, but the risk, and the animal models help us do that. And then comes the first in human studies, which is typically, again, dose-finding safety issue. Even though we can escalate the drugs in the animal models, that doesn't always translate in a one-to-one system to humans. And we have very different, particularly with these immunologic agents, mouse immunology is very different from human immunology. So we really need to understand those in humans, and we start with very low doses to understand what the safety profile is, and then we keep escalating to see what the maximum tolerated dose is. And so you probably have heard about different clinical trial types. We divide them into phase 1, 2, and 3. Phase 1, as I was alluding to, is to find the right dose, side effects, and metabolism of the drug. There's often what's called pharmacokinetics or PKs, and that's where our coordinators are calling you to and get different labs at different time points because we're trying to understand when a drug is given, how quickly does it get cleared by the body. Typically, phase 1s are single arm, although there can be combination phase 1 studies where you're combining different drugs and trying to understand the doses. They're typically small studies, less than 50. Now we move to phase 2. Once a drug safety is better understood, we now need to know how well does it work in myeloma. And we're looking, of course, at additional safety issues. This is typically also single arm, but you can have sometimes randomized or two-arm studies that are phase 2, and that's typically done when you're trying to compare, say, 2 drugs to 3 drugs, but not necessarily the larger phase 2 that we'll talk about. And the sample size or the number of patients in phase 2 can be anywhere from 50 to several hundred, depending on if it's a randomized phase 2. Then and last comes phase 3. This is the definitive efficacy and safety study. And we need 2 arms patients are randomly assigned to receive either experimental therapy or standard therapy. And we'll talk a little bit later, because I did see one of the questions was about the pembrolizumab or the checkpoint inhibitor studies, and that's an important study that we should discuss. And it illustrates why phase 3 studies are important. Just so you understand how FDA looks at these studies, you will not get any FDA approval based on a phase 1 study, because even though you may have a safety understanding a dose, you don't know how well it works. Phase 2s can lead to FDA accelerated or contingent approval if it's an unmet medical need. So the recent examples of that are pomalidomide, carfilzomib, daratumumab. All of those were approved because they worked as a single agent, even in a phase 2 study. In contrast, drugs like elotuzumab, ixazomib required phase 3 studies because they were approved in combination. So it depends on what kind of study, what patient population is being studied. We also have very important other types of trials, longitudinal studies. I think a lot of folks have heard about COMPASS, but don't maybe not understand the huge undertaking that this is. To get complete genomic profiling on the entire patient for over 1,000 patients is tremendously expensive and requires a lot of resources. But that's going to be a gold mine because no other study will do that level of detailed analysis because it's a lot of money. And pharmaceutical companies, while they do need to, they're definitely interested in helping patients. There are also their financial constraints in drug development. So you need nonprofits to do those kinds of studies to really advance the science of why different drugs work in different patients. There's also registry studies, which are not necessarily interventional. They're just being enrolled every patient to see how they're doing. And there's some global ones, and it's fascinating. I was recently in Singapore where a patient can only have Revlimid for 10 months once in their lifetime. You can't reuse it, and it has to be 10 consecutive months. So the physicians are working with their patients to tell them to hoard the bottles and take it every other day. In Mexico, they don't dose carfilzomib by the dose, they dose it by the vial because they don't want to waste drug. And so these are the cost constraints that are occurring globally that really affect drug use. And we are, in the US for now, are temporarily absolved of some of that, but eventually we really need to be thoughtful about how we use drugs. Expanded access programs are really important. When a drug looks promising, this is the way for both the patient and the pharmaceutical company to benefit because, for example, when we had daratumumab most recently, expanded access program, I think we accrued 50 patients in 2 months because there was such an unmet medical need. The drug was looking promising, but it hadn't been quite approved, so the Janssen allowed this drug to be given on a compassionate basis. Of course, it ruined our infusion center because to give that many patients DARA for such long a time really was, but it was in the interest of patients. This slide just shows you what the time frame is like for a typical drug. So we learn the preclinical testing, meaning before we even touch humans in the laboratory, we have those cell line studies and then the animal model studies. That can take up to 4 years. And then the company has to file for what's called an IND, investigational new drug application. And that's the FDA's chance to review that study and verify that it is safe to go into human testing. And then you go through those phase 1, 2, 3 studies, and that can take up to 8 years, 9 years. And then finally, you have post-clinical trials where they can be additional safety. So you're looking at almost 15 years for non-oncologic products. In oncology, we can't wait that long, and the timelines are a little bit more compressed, but this is why we need patients to be participating in studies. In Europe, it's almost the default that patients participate in studies because a lot of access to drug is really severely constrained. In the US, we don't have some of those constraints, but we still need patients to be enrolled in studies to advance our treatments. So what should you do when you're thinking about a clinical trial? Talk to your doctor about your eligibility, meet with the research nurse. We have amazing nursing teams. We may review as physicians at a high level, but they can review in detail what you're concerned about. Our clinical research coordinators can give you what's often most important is what does it mean in a day-to-day basis? How long are your appointments? How often are you coming in? So we have a whole team. It's very important to review the informed consent paperwork. I sit on many committees at our institution, including the IRB, the Institutional Review Board, and our job is to always review our colleagues' studies to make sure they're ethical, sound, make sure that the consent forms are in patient-level language. But at our hospital, we do not allow patients to sign therapeutic studies on the day that they get the consent. We require people, even if you say, I'm ready to sign, you're required to go home and at least read the consent form. I encourage people to share it with their loved ones so that everybody knows what you're getting into. And then if you're still interested, you come back the next day. We don't necessarily do that for studies where you're just donating blood, but I think it's really important that you understand what you're getting into. And it's also important that the consent form be in the patient's native language. Somebody did ask about Spanish-speaking information, and MMRF, MMRC can help with those as well, but all consent forms should be ideally done in the patient's native language so that they understand what they're doing. What are some concerns about clinical trials? So one is, what if I get a placebo? In oncology, in active cancer, it would be unethical to do placebo. The only time we usually do placebo is either 2 drugs versus 3 drugs where we don't know what the value added is, or in perhaps somebody who's in remission or in somebody who's not yet requiring treatment. Those would be the only settings we would do a placebo. It would otherwise be unethical to do no treatment for a patient. Once you start the trial, will you be obligated to continue? We address that in the question. There are always ways to discuss that, but I would strongly encourage that before a patient discontinued that you discuss with the team. Clinical trials are standardized because we need to make sure that patients are treated the same way so we can understand what the side effect profile and the benefit is with a standardized therapy. That does not, however, mean that every patient must stay on the initial treatment dose forever. We can adjust the doses of the drug, hold drugs, change the schedule, work with the sponsor, so there's flexibility in a clinical trial. Will you be treated as a guinea pig? I would argue you're actually getting more attention in a clinical trial because you have a research nurse, the coordinator, a lot more hands and eyes on you. And even though it may be an experimental treatment, you're going to potentially get more attention than you would outside of a clinical trial. As Dr. Martin mentioned, the frequent flyer miles that you're accruing. And clinical trials aren't just for people who've run out of options. You heard from Dr. Gabriel the benefit of RVD induction therapy or initial therapy. If you go back, though, it was thalidomide initial therapy, which was not well tolerated. Then came Revlimid or lenalidomide induction therapy, but that was only 2 drugs. So then we saw that the 3 drugs are better. Now we have frontline studies comparing RVD plus or minus DARA. So even though those patients are going to do very well, this may very well be the next combination that's going to look very promising. We covered, I think, the issue that you always have control, and it is informed consent. So what are the benefits of clinical trial? As I alluded to, the institutional review boards are responsible, along with other committees, to make sure that every study has strict scientific and ethic review. There are certainly going to be normal visits. When you take, for example, a novel medication and add it to the standard of care visits and medications are often billed to insurance because those are routine. Anything that would be investigational is typically covered by the study. As I alluded to, there's a big team involved in research and often higher standard of care. And we also talked about how trials aren't just for patients who are running out of options. And even in earlier stages of disease, today's experimental treatment may be tomorrow's standard of care. So how do you find a clinical trial? And this was one of the questions that had come in earlier as well. So first, you want to ask your hematologist about any available studies. This is important if you can get to an academic medical center. Some of our newer studies, for example, allow you to come once a month to our center, but you can get a lot of the other treatments closer to home. So it's not that every single treatment must be done. However, obviously, it's always on a case-by-case basis. The NCI, the National Cancer Institute, has a clinicaltrials.gov website that you can type in myeloma. You can even type in a stage. You can type in a drug, and it'll pop up with what studies are open, where, which ones are accruing. MMRF, of course, has done a great job bringing the leading academic institutions. We all know each other from all of these conferences because we not only meet here at these kinds of settings, but we're meeting at all of our major academic meetings, coming up with what are the next important studies that are looking promising. And really, that's why this progress is happening quickly. So how do you know if you can participate? Every study is going to have eligibility criteria. At a minimum, they're going to look at your kidney function because when a drug is given, it's typically processed by the kidney and liver. And if somebody's kidney and liver are not working properly, that patient may experience more side effects, but not necessarily related to the drug or the dose, but because of their own medical issues. Low blood counts can be a problem for many clinical trials because, again, some of the new drugs cause lowering of blood counts. So if you're starting off with low counts, the toxicity profile will be very different than somebody who has good blood counts. If there's another type of cancer, usually we're only trying to study one cancer at a time, and when there's a concurrent cancer, the impact of that cancer as well as the treatments from that cancer can confound what we're looking at. Myeloma that is hard to measure. There were some questions actually we didn't cover yet either, which is oligosecretory myeloma. So myeloma we think of as, in the initial discussions, as typically secretory. We can measure the IgG, the M spike, the light chains, the urine protein. However, there's a group of patients who are oligosecretory where they don't meet the necessarily standard clinical trial eligibility, which is typically an M spike of at least 0.5 to 1, a light chain of 100 milligrams per liter or 10 milligrams per deciliter, or urine protein of 200. And the reason for that is when you have very, very small levels of protein, there's going to be a natural ebb and flow. If you did your blood from each side of the arm, you might get an M spike of 0.3 and 0.4. It doesn't mean that the disease is different. And so when you have very low levels of paraprotein or myeloma proteins, it can be very difficult to tell whether it's fluctuation of the disease or the treatment's working. So pretty much all clinical trials will have that requirement. So there's a third group of patients who are truly non-secretory, where there's no evidence of protein by any of those things that we measured. And those patients are typically followed by either radiologic studies like PET scans where there's a plasmacytoma or serial bone marrow biopsies, because the marrow is the only place where you see it. Many clinical trials will exclude those patients because it's going to be quite cumbersome. Patients may not want to get a bone marrow every month like they would their normal blood test, but yet how would they know if the new drug's working? So those are the typical barriers to clinical trial accrual, but they are also surmountable, and we try to have a good portfolio so that we cover all of our bases. A lot of questions about finances, both standard of care and experimental. You may receive medications at no cost. Some of these new drugs are exceedingly expensive, and they would not be covered by insurance. For example, the study I alluded to, daratumumab, if we tried to prescribe that to a newly diagnosed patient, it would likely be declined by the insurance company because it's not yet approved. But that same regimen in a clinical trial, the daratumumab might be covered through the study, or will be covered. Other standard of care is billed to insurance as usual. So that includes marrows, imaging, urine tests, blood tests, because those are part of routine myeloma testing. Many clinical trials will allow money to be given to cover hotel stays, gas, food, airfare. I can tell you from the IRB point of view, this often generates questions because if you pay too much, it's thought to be coercive, and especially patients who may be indigent, that may be perceived as an incentive to participate. So usually these reimbursements are meant to be commensurate with the level of taxation on the patient in terms of what is the inconvenience or frequency. But the clinical research coordinators are excellent at telling you about all of these issues. They can tell you on each study what's covered, what's not. Are there additional resources available? When will you get those reimbursements? What are the important things to know as a subject, which is what actually the term is when we use clinical trial, or as a patient who's participating in a study? You need to tell the study team about everything you're taking, medications, vitamins, supplements, and also ask before starting anything new. Remember that especially for these early phase studies, we don't even know the side effect profile. We don't know what these drugs are doing, and we certainly don't know about drug-drug interactions. So if somebody's taking something, and I don't think this came up, but for example, vitamin C and green tea have been shown in laboratories to kill myeloma, but they've also been shown that potentially they interfere with the proteasome inhibitors. So there's an example of a supplement that many people use but may have some adverse issues with myeloma treatments. I really encourage people to keep diaries of side effects. One of the reasons oncology is a specialty in and of itself, why isn't your internal medicine doctor prescribing all of those drugs? It's because it's what we call a narrow therapeutic index. If you don't give enough, you're not going to kill the cancer. And if you give too much, there's going to be side effects. And that's why this is a specialty in and of itself. But every patient is different. And just because one patient has a penicillin rash, it's not that we're not prescribing it to other patients. The good thing about myeloma is that because our schedules, although this is obviously generally a bad thing, because the schedules are complicated, some drugs are 3 weeks on, some drugs are 1 week off, some drugs are weekly, some drugs are continuous. That on and off can help us tease apart sometimes what side effects you're having. If the side effect goes away during your week off of a pill, that's compelling evidence. So it's good to have a diary of when you are getting treatments, and particularly at Mount Sinai, a lot of our patients aren't necessarily being treated on site. They're being treated in the tri-state area. And so it's really helpful when you come with a journal of what's going on. When did you start that cycle? When did your side effects happen? So we can keep a better eye of that. Bring any records or tests that you're getting at an outside hospital. Particularly on clinical trials, we are obligated to monitor, especially if somebody goes to an emergency room. It could be unrelated to myeloma. It could be some chest pain or asthma, but we still need all of that data to submit when we're reporting patients' outcomes on clinical trials. Keep your appointments. A lot of these are fixed schedules, so it's really helpful to stay on schedule. Tell the team if you have any concerns about this continuing therapy, and obviously always ask questions. So what are some of the new exciting agents in myeloma? And so in no particular order, just more class-based. There's oral proteasome inhibitors. We already have ixazomib, which is the first oral proteasome inhibitor, but two others, aprozomib is being studied, marizumab. There's new IMiDs, so we have thal, len, and pom, amazingly based, again, on that single patient advocate that you heard from Dr. Gobriel. We now have this billion-dollar industry, which now has two more compounds coming out, CC122, which is a next generation. These are all sometimes called cereblon-binding agents, which is the target protein. We heard about histone deacetylase inhibitor like panovinostat. There's a specific one called HDAC6 inhibitor called ricolinostat. Then this next class of drug called kinase inhibitors are approved in many, many malignancies, not yet always in myeloma, but vemurafenib is targeting a particular protein mutation, BRAF, RAS, afusiratib, it's AKT3, dinocyclib is cyclin D1, ibrutinib is a BTK inhibitor, trametinib, RAS, dabrafenib, so on and so forth. Some of those, they don't have a name yet. That means they're very early in development, so typically not yet approved. Isatuximab, you heard from Dr. Martin, which is the next CD38 monoclonal antibody. There's some antibodies that are attempting to also overcome patients with P53 deletions, which is really a high-risk unmet medical need. You heard about venetoclax. Selinexor is a really interesting, it's an oral medication. It's a nuclear transport inhibitor that prevents proteins from moving out of the nucleus and potentially can help, particularly in high-risk patients. Felanicid is an intravenous kinesin spindle protein inhibitor. And last, you've heard a lot from my colleagues about the immune checkpoint inhibitors. And maybe in the Q&A period, we can talk about the studies. And, of course, the immunologic therapies like CAR-T, BiTE. So this just, I think I'm going to just not spend too much time on this because you've seen slides of this. But on the left is the monoclonal antibodies, drugs like DARA, which are just given intravenously, and they work. Sometimes they also have a payload or a conjugate to deliver toxins along with the antibody itself. There's also CAR-Ts, which are you've heard about from Dr. Martin, and the vaccines are also being studied. I think the slide that I had somehow didn't make it in here, but the bispecifics are also very interesting. So let's see. Yeah, that's not there. So the CAR-T product, you have to basically, this was a question that came up with the stem cell discussion. Stem cells collected for autotransplant are completely different from the CAR-T product, so we're talking about different things. CAR-Ts, the cells are taken out of the body, the T cells, and they're genetically modified and reintroduced. And the limitation of that, there's some time required for that production. These new bispecific antibodies can basically take an antibody construct like DARA, ELO, but then at the other warhead, they can bring T cells into proximation. So it's kind of like getting CAR-T benefit, but with an off-the-shelf antibody that's ready to go. So we have very exciting clinical trials with both that class of drug as well as CAR-T at Sinai, and I'm sure our collaborating institutions also have similar compounds. And some of those are actually listed here on the monoclonal antibodies. A lot of the ones that are numbers, the GSK, the J&J, the Pfizer, those are all these bispecific antibodies that are very exciting, and a lot of small molecules in development as well. You heard about these studies. There's also a lot of work, as Dr. Gabriel mentioned, in smoldering. Do we really need to wait for those crab symptoms to develop, or can we intervene early? But we also need to do a better job of making sure we pick the right patients for that. So to summarize, I think research has brought us to where we are. We don't expect anybody to be a guinea pig. And all clinical trials have exceedingly tight supervision and standards. They're scrutinized by the FDA. And we really need close communication between patients and caregivers. So thank you. And at this time, so I'm seeing some questions. Maybe this is an opportunity to talk about the Pembro study. So the pembrolizumab is a checkpoint inhibitor, and we haven't talked about this in detail yet, but basically cancer cells have a cloak to protect themselves from being attacked by the immune system. And that cloak is mediated by two proteins called PD-1 or PD-L1. We have agents that remove the cloak, and now these antibodies allow the immune system to go after the cancer. So this decloaking mechanism, this checkpoint inhibitors with PD-1, PD-L1, has many different drugs approved for many different cancers, solid tumors, and also Hodgkin's lymphoma. So there was a lot of interest in this in myeloma. In the initial phase 2 studies, there were even patients who had response with either Revlimid or pomalidomide where they didn't respond, but the addition of these checkpoints made them resensitized to the disease. The problem was that, obviously, that was a single-arm study, and this is a great example of why we need clinical trials. So phase 3 studies were done where everybody got Revlimid and Dex, half got the addition of the Pembro, or everybody got pomalidomide and Dex, and half got the Pembro. And so surprisingly, even though there was that initial signal of potential benefit, both studies had to be halted because there was no apparent improvement in outcomes and actually an increased risk of toxicity, and specifically deaths, in both studies that led to the closure of those studies. And it's a great example of why we need clinical trials, because something can look promising in phase 1, look promising in phase 2, be approved in many other tumors, but until you do the combination study and do the proof of concept, you really don't know what it's adding. And I think, so this is obviously given pause, and so again, this was tightly scrutinized. These studies were not allowed to continue. There were interim analyses that basically halted all studies. Not only were those studies halted, every myeloma study using this combination of an immunomodulatory drug like Revlimid or pomalidomide was halted. And on a case-by-case basis, each study and physician investigator has to appeal to the FDA about whether this particular study. I think what we've learned from that is it is unlikely that those checkpoints will be continuing in early stages of myeloma, because we need to understand better in heavily pretreated patients where there's clearly a more unmet medical need, and we need to better understand that risk-benefit. There are several other phase 3 studies with those agents in myeloma that have not yet seen that signal. So it may be a drug-specific issue, a combination, dosing, schedule, all of those things need to be cleaned up. But that's an important study that came up. I'm seeing here also some questions about, I think we've covered about the health insurance. I hope that was sufficient, but at the end of the day, for a given study, you always have the option of asking the research team, what exactly are my expected costs from this study, what can I expect, and what's standard of care and what's not. Trial and study are synonymous. So CAR T, why is there so little availability? That's an important issue that we're struggling with. Phase 1 studies, by their nature, we're trying to understand the dose and safety. And as you heard from Dr. Martin, some of the other diseases, like in ALL, there were severe toxicities. And so we can't just open up the floodgates and treat hundreds and hundreds of people without knowing the full side effect profile. So all of these CAR Ts, they're being approached from different countries. The US one is the Bluebird, that's quite far advanced using BCMA. Chinese group, Legend Biotech, has another one. The data are very impressive, as Dr. Martin alluded to. These are patients in the U.S. study who had 7 lines of prior therapy and were pretty much refractory to many of the drug class. We have several of those at our institution, and this infusion of the CAR T product led to dramatic remissions with very high response rates. We need more follow-up for the durability, but the benefit seems to be so far very encouraging with manageable cytokine release syndrome. But just even with Bluebird, there's changes in the dosing. The amount of patients that are being dosed with different levels of CAR T product are still what's the right dose. When should we intervene with the drugs to manage the infusion? So I think it's still early in development, and we don't know the toxicity profile yet, who's the right patient for it, how to predict those toxicities. But I think what's nice is that there are these monoclonal antibodies, these bispecifics, that are trying to do the same thing, and they're off the shelf and ready to go. So those are also in phase 1 early development, and those are also available at our institution. So if somebody's really interested, at our institution, we're going to basically do, if a product is ready to go and the patient can't wait for a CAR T slot, they would go into those antibodies. If they can wait for a CAR T and there's a slot ready, so be it.